Hello, good afternoon, everybody. Welcome for coming here. This is Sara Bonella, the Deputy Director, and I'm Ignacio Pago-Navarraga, the CICAM Director. And it's a pleasure to welcome to this first webinar of this series on the challenges and responses in simulation, modeling, and beyond. I hope you and your relative are doing well, and that also professionally you are managing to cope with these difficult times we have to go through. During this period at CICAM, we have thought that it could be appropriate to accompany the broad communities that shape us by creating this format to promote broad interdisciplinary dissemination of scientific activities, challenges, and opportunities that are associated to COVID. We hope that by starting this series of online meetings, we will contribute to promote scientific exchange and communication, and hopefully encourage new scientific collaborations, even if everybody is attending from the distance. Actually, this type of remote meeting is a new format for us at CICAM. We will see how fruitful it is, but then in any case, we hope it proves useful to all of you during this time. We have organized a series in principle of 3 sessions with the idea of addressing from a very broad perspective and also to a broad communities what are scientific issues, challenges, and ongoing initiatives to understand and also to fight this type of viruses and the associated pandemics. So in the spirit of CICAM, we try to keep these meetings live. So that's why we have organized them not too long. Each of these sessions will contain typically 2 short presentations from experts with complementary views or aspects. And at the end of these 2 presentations, we will have a joint session for questions because that will allow that you can address specific questions to any of them, but also you can pose more general questions that can also lead to a dialogue between our experts here. So we will start with these 2 half-an-hour presentations, and then during them, you can pose your questions as it goes. Actually, if you have an account in YouTube or Gmail, you can directly type your question on the site of your browser, but you can also alternatively send your question by email to the address that is in the email that you have received for this session. The email address is director@cicam.org. Basically, then Sarah and myself will collect all the questions and we will relay them in that session at the end to Eric and Francesco, and I hope it will lead to a dynamic and an open also session of discussion after the presentations. So let me then start. Let's move on the program. So the first speaker, as you have seen, will be Professor Eric Lindahl from Stockholm University, who will be talking about proteins associated to COVID and how we can take advantage of how know and manipulate them appropriately. So Eric, please, now the floor is yours.
Thank you so much, Ignacio. Let's see, share a screen first. Desktop. And there we go. Now you should see my title slide at least, right? Thank you so much for having me, and great to, I was about to say, see everyone. Unfortunately, I can't see you, but it's an interesting exercise and a new way of communicating scientifically, and hopefully this can lead to some good things too, in particular that we can interact more without necessarily spending all those hours in airports. For me, at least, that has been the one big upside of this change recently. In one way, I'm not sure I'm a good person to present here, because caveat emptor, I'm neither a virologist, epidemiologist, I'm not an MD, I am certainly not a group that has a ton of experience of drug design, but we are interested in molecular biology, we're highly interested in structure, and of course, we can't keep our fingers away from this on the one hand, in the interest of understanding what's happening to the world, viruses and proteins in particular. So when I presented this to my group roughly 10 days ago, I figured I had a presentation about this already. So when Ignacio asked, I figured I could share this with you too. So in a way, I see this more as a way of kicking off the discussions, and I might very well answer, I don't know, to a whole lot of your questions, but we'll take it from here and see where we go. So what I'm going to tell you about is really, I'm going to start by focusing on the, not so much the disease, but the virus itself, so SARS-CoV-2. And as many of you probably know, or almost certainly know by now, this is a virus that is highly similar to the original SARS virus. It has some 85–86% sequence identity, so very similar. It has a reasonable similarity to the Middle Eastern respiratory syndrome virus too, and then in general, a number of other human coronaviruses. It's a beta coronavirus and a single-stranded RNA virus. If we start looking slightly more into detail, what happens with the sequence? This is expressed as a handful of reading frames, which is possibly a bit different from what you might know from undergraduate bioinformatics or so. There are some 30,000 base pairs, so it's a reasonably large genome for a virus, but these open reading frames are expressed as polypeptides or polyproteins. So it's expressed as one long sequence, and then once this has been expressed, you have parts of this open reading frame itself acts like a protease that cuts up the long reading frame, the polyprotein, into a number of both structural functional proteins and a couple of non-structural proteins. I figured for better or worse, even if we've never heard of this before, the good thing is that there are only a handful of proteins here. The really bad news is that there are only a handful of proteins here because we don't really have that many different things to target this on. What you see here already in blue is which is PL-Pro, and then in cyan 3CL. Those are the main proteases that have the role of cutting everything up into the smaller ones. I'll get back to that in a second. And the yellow part in S here is the spike protein. If we instead look at this from a structural point of view, to the left, this spike protein is what you see here in every single rendering you've seen of the coronavirus. These are the things sticking out on the top, and it's very characteristic of this protein. And for obvious reasons, a whole lot of the attention, both in terms of drug design, potential vaccine development, and everything has focused on the spike protein. There are a number of other proteins. The one called E protein here is an envelope protein. There are a few membrane proteins, and there are some proteins that protect the RNA genome inside it. And the reason why it's so efficient to wash your hands is that this envelope is a lipid envelope. So if a little bit of soap will destroy the envelope here. The virus infects the human cell by the spike protein binding to the ACE2 receptor, the angiotensin converting enzyme 2 on the surface of your cells. And what then happens, to make this a bit simplified, well, grossly oversimplified, is that once this is bound, the spike protein will be cleaved off. So they will basically be recognized by your host cell. This is a bit complicated in itself because it likely depends a whole lot on the glycosylation of the spike protein, which is important because you will, if you just download a structure from the PDB, you will not see all those glycans. And then exactly what happens, that there are people that are far more talented than I when it comes to understanding the entire viral influenza fusion here, not influenza, the coronavirus fusion and how it actually gets into the host cell. But obviously, this is going to be one route towards targeting this protein. Can you prevent this from binding to the AC2, or can you prevent the actual cleavage, or can you somehow possibly target the spike protein with antibodies or something? Not entirely surprising. The spike protein was one of the first that a number of groups focused on getting structures of. There were already structures of this for the original SARS coronavirus. And the Jason McClellan group at UT Austin determined a cryo-EM structure in mid-March that was published in Science. It's a reasonably good structure, 3.5 angstrom. There are two different conformations of the binding domain. They're up in green. And I'm not going to go through all the details of this protein here, but we have a reasonably good idea what this looks like. And we also have a reasonable idea what the whole dimerization interface looks like. And as you see here, that in the lower middle here that it's relatively, I would say more than relatively, it's highly similar to the original SARS coronavirus. Now, as we mentioned before, this will bind to the ACE2 enzyme. And just a week or two later, there was the Xiao lab from Westlake in Xi'an. Managed to determine a cryo structure of the complex between the spike protein and the ACE2. So here you see the ACE2 binding domain up there in yellow or orange, whatever we call it. And this too is a very nice structure. Overall, it's just below 3 angstrom, and even in this most difficult interface to the RBD domain, it's just about 3, might be 3.4 or 3.5. And I think that's one of the big changes. And I have to confess I'm a bit biased here because we are deeply involved in cryo nowadays. But I think it's amazing how quickly some of these structures have appeared. In some cases, you're talking about 2 weeks from the time they purified the protein until they had a structure and were able to submit it. And compared to 5-10 years ago, that's likely one of the big changes, that cryo doesn't necessarily allow us to get super high resolution structure, but they certainly enable us to get them very quickly. Now, having seen this, it might seem obvious that if we understand the spike protein and we understand the binding interface, couldn't one create just an antibody to identify the spike protein or something and thereby kickstart your immune defense? And one of the things that I've sadly learned myself here, and possibly one of the memento moris I want to convey here, that it's important to think of the biology here. I think we're all, or at least in particular me, we're frequently structural biologists or computational people and tend to focus on one small binding region or one free energy or something, but there are some very important lessons here. So if you look at the original SARS virus, people have actually done that. There are antibodies that bind. But the horrible thing that happens is that this makes the infection worse. So if you administer immunoglobulins that are anti-spike, what actually happens is that they create much worse lung injury. So somehow it causes the macrophages, to pretty much take up the entire virus instead. And exactly why is probably not yet known. But the whole point, and you can certainly argue here that there might be some skewed in the statistics here that whether people that were actually the victims that were deceased, they might have had a worse infection and everything. But it's certainly not obvious that just because you can't create an antibody that enables you to treat the disease. In some cases, it can be far worse. So I think it's super important to think of this in 3 or even 4 stages. We can understand binding, whether that is from computational aided drug design or even just high throughput screening. But from there, you have to go to cell lines. And even when you're in cell lines, going from cell lines to actual animal models is a completely different step in turn. So remember that most of these things we read are very preliminary data and simplified assays or something, and that's not necessarily the same thing as having a drug that will work. There are a number of other proteins, and in the interest of time, I'm going to have to go through some of these a little bit quick. The envelope protein is a pretty neat membrane protein. It's one that my colleagues in Stockholm, Lucy Delabot in particular, has been a bit interested in. This is likely a channel for calcium ions, at least on the original SARS. There is an NMR structure that was determined some 2-3 years ago. It's probably one of the proteins that people have expressed less interest in, but one potential use of this protein, even though we don't know exactly what it's doing, it appears that this protein is essential to the virus life cycle, as is most of them. And if you could express a virus without the protein, this could potentially be an interesting vaccine candidate because it will likely not be able to infect your cells, but you can have everything else on the surface of it that will hopefully elicit an antibody generation in your body. The big target that most, if not all of us, have likely seen is the so-called main protease. So the main protease, the reason why this is called the main protease, this is the real workhorse that cuts up this polyprotein into the constituent small proteins. There are a number of awesome PDB structures of this. I won't have time to go through all of them. Here, it's important to be aware of a couple of things. First, it's a dimer, and second, we know reasonably well where the active site is. So there are going to be lots of structures out there that are either monomers or that are, some of them are apo and others are holo structures. And there are important differences here. If you're going to try to design the well, if you want to screen things for drugs, you likely want a dimer that is a holo structure, not a monomer that's an apo structure. I will jump straight to the ligated version of this that appeared a few weeks ago, I think early April 8 or 9, which is Xi Rao and Haitao Yang labs from Shanghai Tech. They determined a beautiful high resolution X-ray structure just above 2 angstrom resolution. So not only is this a dimer, but it's also a dimer where they have structures bound of a particular inhibitor called N3 that they had identified to be an inhibitor already of the SARS-1 protein. And here we see it bound to SARS-CoV-2. You can have, let's see, yes, I do have a second slide here where we show that it's binding and how it's binding, in particular in the active site. So what this inhibitor does is prevents the main protease from binding to the cleavage site and splitting everything up. So in theory, at least, this should be a really efficient way of preventing the virus life cycle. There are a couple of important points here. As computational people, we frequently jump straight into things. And as I mentioned before, we ignore the literature. We all want to help here, but I think it's important to be aware of what has already been done. And so this is an example, not primarily of a computational group, but this group doing the structure biology here did the computer-aided drug design too. They were able to identify both inhibitors that have been proven to bind the SARS-CoV-1. They worked here too. And not only did they screen through fairly large databases, they also assayed some 10,000 inhibitors experimentally, mostly through a FRET probe, so that they can see whether something is bound or whether it's possible for the probe to access this site. And given that this has already been done, I'm not saying that it's useless to start a new study where you try to screen things to the main protease, but if you're doing that now, I think it's important to have some sort of strategy. Why am I doing this, and what is that I can add that has not already been done here? And I'm not even sure whether I know that answer myself for some of these proteins. So in this particular case, they were able to identify a number of hits. They weren't able to identify a number of hits that didn't work, but they were also able to identify a handful of hits that were quite interesting. And apart from this N3 inhibitor that was already known for SARS-CoV-1, there's a compound called Epsilon that is not FDA approved actually, but it's undergoing early clinical trials. So it will likely be possible to hopefully get that approved reasonably soon. And not only do you get good inhibition here in the simplified assays, as you have up on the left here, but you're also able to show that this works in vero cells. So that's kidney epithelial cells from African green monkeys. And as you see there up on the right, you actually lead to a significant reduction in the real-time PCR response, both from Epsilon and N3. But again, this is still simplified cell line examples. But I would say that there are some reasonably good indications that there are inhibitors that could work against the main protease. The other thing that, I'll come back to this in a second, the other thing these studies enabled is that Diamond were able to take the structure and determine an even higher resolution. I think it was 1.2, 1.3 angstrom X-ray structure of the unligated form. And they also have fragment screens now. I think there are 66 total small fragments bound out of 1600 attempted, both covalent and non-covalently bound small fragments. And you can download all of this. They're available both in the PDB and this link, Diamond, below. But even that might not necessarily be enough. So just to highlight one more study, the Hilgenfeldt lab in Lübeck, they published a study just mere days later for slightly different types of inhibitors. I'm not skilled enough to determine which one of these is better. But I think the interesting things you can find in this paper in particular is that they start to discuss the whole ADME-tox process. So what is the average lifetime, the mean residence times of these various compounds in tissue? Can you actually deliver them through inhaled inhalation or something? And here too, it's exceptionally early, but this, again, this is also something that all those of us who are computational, me included, frequently ignore. It's not enough to have something that's a strong inhibitor. It also has to be something that has a realistic chance of actually being delivered to patients. And these are roughly, well, around 1 micromolar affinity binders. The other thing that appeared about a week ago is the structure one of these so-called non-structural proteins, which is an RNA-dependent RNA polymerase, long name. This is, well, it's an RNA polymerase, and I will not go through the entire structure here, but the one important part here is that this is the likely target of the remdesivir compound that you might have heard a bit about in the news. And based on previous findings of remdesivir to other RNA polymerases, in particular for herpes viruses, you see if you use this red domain that's called palm. If you look at the upper left here, in the upper part of the red domain, roughly where it says fingers in blue, there you have a small binding site where this remdesivir is likely to bind, based on modeling this in on the HCV viruses. And that's what you see here on the left. So here we see both remdesivir and another compound called sofosbuvir, me in Latin. Both of these are nucleotide analogs and appear to be very strong and good inhibitors that, again, they appear promising, but here too is the fundamental difference between, if you just looked at this from a computational point of view, these are so great that you would imagine that if we just administer a bit of this, it would completely obliterate the virus instantly. And there are interesting signs that remdesivir might help a bit with these infections, but it's certainly not 100% that all patients immediately get cured. So here too, with a bit of luck, we might know in a month or two if remdesivir is good. Of the drugs I've heard of and been aware of, this is likely the most promising candidate. So if you want to try this, this is a slightly different challenge than we do traditionally for drug design. So normally you would use these gigantic libraries of everything available. The problem with that is that it's going to take a year or two before anything is clinically approved. So what most people in the world are doing, including the collaborations that I'm involved in, others, they tend to use some sort of either repurposing libraries or libraries with drugs that are known to be safe in man and that are in some sort of early clinical trials. There are a handful of different of these. There's a broad repurposing library. We tend to use a version of that from Fraunhofer, but it's essentially broad. Dompé, an Italian pharmaceutical company, we have access to their library, which is pretty much a collection of Integrity, Drug Bank, and Drug Central. And as you see here, that there is some reasonable overlap here, but most of these libraries, they're not identical, and you might want to try several one of them to see if you find something interesting. The sad part here is also that this is where it might be a long shot. If we find something that's good, but that's not in these libraries, the likelihood that you will get it approved even for some sort of emergency usage is unfortunately very low. But there are people that are far more talented on that than I am. The other thing that I would strongly suggest that you do is that if you're starting drug design here, you might have an awesome method. Congratulations, that's far more than I do. But if you have such a great method, and if you're now approaching these companies and arguing that other people should start determining, well, doing experimental studies for you, start by cross-checking. Are you able, or is your method able to predict the actual inhibitors of SARS-CoV-1? Because this is 86% sequence identity. If you're not able to identify even qualitatively the binders or inhibitors of SARS-CoV-1, I think it's sadly highly unlikely that you will be able to do it for unknown data for SARS-CoV-2. There are a couple of papers out there. I like this particular one in Journal of Medicinal Chemistry about a year or two ago, and they've assembled data from the 3,000–4,000 publications out there and identify some 122 inhibitors and peptide mimetics in total. So that's, I'm not saying that you have to identify all of those, but you should be able to pick out a handful of those in a larger data set if you trust your method a lot. I think that's roughly what I'm going to tell you about the proteins here and roughly what I know about them. I'm going to tell the other part that we and a few other teams have been involved in is try to establish some community resources to avoid everyone having to bootstrap this from scratch. So in no particular order, and I'm sorry, I am sure that I'm missing some things here. That is not intentional, but it's simply lack of time. The Global Health Drug Discovery Institute is a China-based organization supported by Bill and Melinda Gates Foundation, that's Tsinghua University, and they also have support from the Beijing municipal government. And they have collected a number of information about publication data, preclinical and clinical trials, data analytics, and a bit of structural modeling, and also computer-aided drug design. And this was one of the first studies out there, and it's certainly worth browsing through this before you're starting to do things yourself. The opposite is something that appeared literally yesterday, that the European Bioinformatics Institute, together with EOSC-Life, so the European Open Science Cloud, they launched a COVID-19 data portal, covid19dataportal.org, where they focus primarily on experimental data. So you have a ton of sequence information here, you have all the expression data, a bunch of literature. You don't really have a lot of original computational data for now. We're working with them and possibly having this as a hub where we can also upload very large amounts of, say, computational screening data if you want to train AI methods or something, but that's not quite ready yet, so don't blame them. The advantage here is that EOSC and EBI, they have very large storage infrastructures that we can hopefully use. But then there are people like us, Chekam and others, and that's certainly where my team falls too. And that's difficult because, of course, computational data is not original in the same sense that is a PDB structure, but I still think it's important that we start acting like a community and share data. So what we've started to do there is that a number of teams here started to develop data sites, and then we realized it's just stupid to have half a dozen of orthogonal data sites, or worse, partly overlapping. So the Molecular Sciences Software Institute in the US and BioXL in the EU and a number of other teams, they joined forces here and tried to create a site focused a bit more on the stuff that computational groups such as us are interested in, but we don't want to limit it only to computational data. So we've also tried to include things like, well, PDB structures, models, reasonable targets, therapeutics and databases, simulations and assays, anything we want. And the idea here is that we are going to be more liberal than some of the other sites. If you do anything that's useful, you can directly go either here or to Zenodo, upload your data, and 50 gigs, you will not need any permissions whatsoever. You can immediately get a DOI and add your data here right away. And the idea here is we really believe in sharing early and sharing often. There will be things here that are mistakes, and mistakes are fine. So we're experimenting with some sort of quality control where we're going to try to grade things. When they're initially uploaded, we don't say anything about it because it's unknown. And when there is enough data that we can at least try to assess the quality, we're going to mark it as yellow. And when there's data that at least one of us had looked at it and find that this might be useful to somebody else, we're going to try to mark it as green. That doesn't mean that non-yellow or non-green data is less important. It's just that see it as some sort of real-time, very lightweight, I wouldn't call it peer review, but that we try to screen things at least and point out obvious mistakes. I'm going to show you a demo of this in 1 second, but I want to mention two other initiatives that could be worth talking to. In the EU, we have a site called Escalate for Coronavirus, which is a typical European Union project with a number of teams have teamed up, led by Dompe in Italy. So here you have a number of simulation and docking groups involved, but also medicinal chemistry groups, pharmaceutical companies, and people focusing on assays. And they will be able to help with testing here. One thing what's not quite clear yet is that how we're going to help funnel all the information and contributions from the community, because it's way harder to test things experimentally than it is to make computational predictions. So for now, don't take that as a lack of interest. It's just the experimental groups, we jointly need to come up with a way to prioritize predictions for the experimental groups. There is at least one similar project in the US, the COVID moonshot project from the Postero.ai, where a number of colleagues of us are involved in. And overall, it's largely the same setup, slightly looser, but here too, you have a number of computational groups that try to make predictions. These predictions are then forwarded to experimental groups that will try to assay these predictions. And based on the outcome of these assays, we will then try to improve, or they will try to improve things in the next round. These are by no means exclusive or anything, but I think it's important for all of these projects to start talking. The problem here is that there's too much to do and too few people. So in anything, we should try our best to avoid duplicating efforts that other people are already doing. And in particular, don't start working on the main protease, because that's probably what most other people are doing. I'm going to see if I can quickly give you a small demo of one of these sites. Yes, you might be able to see my screen here. So this is the MOLSI BioXL site. So what we've tried to do here, in particular, thanks to tons of suggestion by John Shodera, you can, for instance, look here at the targets. So we can look at the main protease activity, and then there are a couple of entries here that what are the potential therapeutic modalities. So we can try to approach with small molecules in particular. You can list all the proteins. In this case, particularly, it's reasonably easy because that was one specific protein I looked at. You can also look at what therapeutics might be related to this. So if I click the RNA-dependent RNA polymerase, I would likely find remdesivir. But you can also find a lot of resources here. So for instance, if you want models of all the things that have not been determined yet, you have a ton of example models here. For instance, Michael Feig's group has done a ton of things where they've taken the AlphaFold predictions and then tried to improve them further with molecular simulations. And I think that there's some of the best models you can get there right now. And at least for me, it's awesome because that means that I don't have to reinvent the wheel. I can build on the shoulders of giants. You can also go to simulations here. So rather than me trying to repeat the simulations for the main protease here, we can, for instance, download the 100 microsecond MD simulations that DHO has already done and shared. So rather than me running 1 microsecond simulation, I can instantly start out with a factor of 100 more data. And there are more collections coming from DHO, and we're also going to have Folding at Home share all their Macrostates models of this shortly. And the interest of time here, I'm going to have to continue. So just as one example, what Catherine Berg in my group did in roughly 1 day is that she downloaded these the main protease simulations from the site. And rather than running her own then she did principal component analysis, first in Cartesian coordinates, and then she built Macrostate models of these. And now we're running out of time here, but that's fine. So this particular case, we identified a number of different clusters, but they look somewhat horrible, to be honest. Strange timescales, and the timescales are, well, the states are not clean in that sense that we don't move from one state to another state and then stay there. You can try to clean that up by doing 4 or 5 dimensional clustering with time-independent component analysis. And then eventually we find a number of macrostates from these, and these will be shared at least on one of the sites. In this particular case, Catherine wasn't super happy with these states, so then she tried something else and clustered this on with models based on elastic network modes like distances, so pretty much just accounting for distances within a relatively short cutoff. And here you end up with slightly more fine-grained states, like at least 4 states or so, and they might have slightly better kinetic appearance, although it's certainly not beautifully converged yet. And here we actually get 4 nice macrostates, and in particular, we see reasonably nice motions in the main protease where it appears to move between conformations corresponding to the apo and holo state, although we started from the apo state here. So these are, of course, not authoritative, and there are likely better analysis by now because this is 10 days old. But I can, if there's one thing that I want to suggest, use the resources already out there. And when you get resources, try to upload them again. Nobody's going to complain that your data is not perfect. Somebody might point out that there is a mistake here, so we probably shouldn't share this until it's fixed. That's fine. But the one thing that worries me more than ever is that we're all reinventing the wheel here and starting with the vanilla simulation of the main protease, which is likely oversubscribed by a factor of 25, while there are a ton of other proteins that nobody's looking at. So in summary here, what I think is going to happen in the near future here, many of these consortia, and well, many of you listening, there are people publishing ranked docking hits for several targets. Not just ranked docking hits, as we see in the literature. Some of them already have experimental assays. So you're going to have fairly good inhibitor candidates. Many of them are also based on structures. You will have co-crystals or even inhibitors seen in cryo-EM structures. There will likely be a lot of groups trying to relax these poses, possibly even calculate free energies for them. That's great in some ways, but we're still stuck in this very simplistic binding things. And whether that's going to contribute to things that actually have therapeutic effect, we have no idea whatsoever. There are likely going to be a number of groups that attempt docking both to APA and holo structures. Overall, I would say many of these things are likely useful to understanding the power of the proteins and the power of these methods in general. I think it's less clear whether it's going to be able to contribute to any therapeutics that are actually curing COVID-19, sadly. And to be a bit humble here, although we are computational groups, if there is anything that's my take-home message here, read the experimental studies, because I think the experimentalists are frequently significantly further along than some of the those of us who are computational groups believe. That does not mean that the computations are useless. On the contrary, but it's there are tons of things worth doing because we're interested in the SARS-CoV-2, but that is not necessarily the same thing as the things we can do that actually have a chance to influence the outcome of drug design or therapeutics. And the final take-home message here is collaborate. Find something that other people are not doing and make a shout out at one of these sites that I'm interested in X. See whether other groups are also interested in X, and in that case, join forces instead of having 4 groups working on X in parallel. And with that, I think I've used my time, so I'm going to stop there. Thank you.
Okay, Eric, thank you very much for this very illuminating presentation to start with. As I said at the beginning, now we will stop for 15 minutes so we can refresh ourselves. And I will invite you to come back at 5 to 4 to listen to the second presentation by Francesco Stellacci. I wanted to mention, because I forgot at the very beginning in the introduction, to say that this is being streamed live, but the presentations are recorded and will be kept in the CCAM website. So you can always come back later to check or to revisit any of the presentation or the question session at the end. So thank you very much, Eric, again, and see you back in 15 minutes. Hello, everybody. Welcome back to the second part of today's webinar. As I explained earlier, we will start by a presentation by Professor Francesco Stellazzi from the EPFL, who will be talking about new routes in the design of antiviral drugs. So, and afterwards we will have the common question and answer session. I remind you that you can post your questions during Francesco's presentation, both to Francesco and to Eric's talk, directly on the YouTube webpage, if you have an account in YouTube or Gmail, or you can email your question to director@ccam.org, and we will just get all the questions that we receive from you, and Sarah and myself, we will be relaying them to Eric and Francesco. So please, Francesco, now the floor is yours.
Hi, everybody. Thanks for this opportunity. I'm happy to present to you my research, and I should start by saying that I will not talk about coronavirus. So you heard a lot in the previous talk, very interesting for me personally, about the specificity of coronavirus. Today you will hear, no, in the next half an hour, you will hear what I have been doing in my laboratory in the last 10 years in working in antivirals in with an angle that is a bit different from many others, given my background, that is that of a material scientist. So, and I'm an experimentalist. So why antivirals? You know, in these days, it looks like almost a known thing that we should work in antivirals, but that's not always obvious to many people. And for me, the answer is in the fact that viruses kill and are responsible for a lot of key diseases in developing countries, and they kill millions of people every year, and in many cases we really do not have a way of fighting them. How do you fight a virus? Well, the best way is using a vaccine, but as you can see from this slide here, we don't have vaccines against many viruses. The other approach, once you're sick, whether or not a vaccine exists, is to use a drug, therapeutics. And we have two very good drugs against viruses, the cocktail against HIV, and we have also anti-papilloma virus, anti-hepatitis C virus drugs. But but there is a lot of viruses against which we don't have any drugs. Now, as you've also seen in these days, when we talk about viruses, we don't simply talk about the viruses we know, but we also have to be careful about emergent viruses, emerging or re-emerging viruses. Now there is a number that I often quote in these days, and that is that starting the 70s, there has been a new virus that has emerged roughly every 4 years. If you look at this slide here, there are way more. I'm obviously talking only about non-influenza virus, and I'm talking about major viruses. And when a virus emerges, you never know how hard it will hit. So if you look at this slide here, this is the average lifespan of Americans in the last century. As humanity goes, we've done very well. If this was European, it would have started slightly higher, but the concept is the same. And this dip right here is the Spanish flu. The broader dip here is First World War, and this dip here is Second World War. Now you see how bad a virus can hit when it emerges. Now I don't think coronavirus will do that big damage that the Spanish flu did for many reasons. But if you will in future plot world economy, you will see coronavirus in a plot forever. So there is a lot that a virus can do when it appears and it emerges. So put together the two main concepts that I've done so far. On one hand, I said that we need many drugs to fight many viruses that hit underdeveloped countries. On the other hand, I said that we need drugs that fight viruses we don't know yet. So viruses that we know for sure will emerge. We cannot hide that. How do you put these two things together? Well, in my mind, the way to put these things together is to start working and to work hard in developing broad spectrum antivirals. So drugs that can actually fight many viruses in developing countries, at the same time that can be ready, and I stress this, ready when a pandemic arrives, or an epidemic, doesn't need to be pandemic. Now, if you want to develop that, you have to look broadly and think broadly, how do we fight viruses? Now, typically we divide antiviral drugs in various categories, and the boundaries are very blurred. So these are things that you have to take with a grain of salt. The first category, and these are all basically all FDA approved drug, it's antivirals themselves. If you look at the virus replication mechanism, well, in a virus replication mechanism, the antivirals work intracellularly and they prevent viral replication. For example, in the previous talk, when we were talking about inhibitors of RNase and other enzymes, those are antivirals classically. There is another way of fighting viruses, and the other way is the same way, if you want, we fight most bacterial infection. That is to say, I come to a virus and I destroy it. Or if I want to be scientific, I irreversibly inhibit its infection. Now, this is called virucidal approach. It makes sense if you go biologic, but unfortunately, if you think about the virus replication mechanism, chemically a virus is made of biologically, of the same entities that we are made of. And so, unfortunately, what chemically damages a virus also damages us. And so I say, you know, in these days, we know that soap can actually damage a virus. Strong acids are what we typically use on top of soaps in the labs to clean, but also pure alcohol. We've heard a lot about alcoholic solution in these days. So before coronavirus, I would always make a joke that when I have an influenza, I try this approach. It doesn't work, but it makes me happy. But why doesn't it work? Because all of these things not only damage the virus, but also damage the host cell. So they are good extra host ways of disinfecting, but they're not translatable as drugs. There is another approach to fighting viruses, and that is also extracellular, and that goes in a category that we can call broadly as entry inhibitors. That is, these are compounds that typically, when they work on a virus, they will bind to a virus and prevent viral entry into the cell. This goes, for example, in the previous talk, to most antibodies that are nowadays being sought to bind to the S proteins. They will actually act as entry inhibitors, if I understand that correctly. Now, the problem with this approach is that upon this is based on binding, and binding is a reversible effect, and upon dilution, the virus compound complex is actually dissociated, and an infective virus is released, and this infecting virus can restart the viral replication mechanism. So for the most part, these approaches have actually failed to be translated into effective drugs. There is now some monoclonal antibodies that prophylactically seem to work on against RSV, but one has to be cautious there. So this is the landscape. But when I look at this landscape, I have to say that these particular classes of drugs are actually very promising, if not for the reversibility effect. Why are they promising? Well, they're promising because to find compounds that bind to viruses is not so difficult. In fact, viruses, for the most part, all have a conserved attachment ligand, so a ligand that binds to a cell membrane. This conserved attachment ligand is conserved for many reasons, but I can say that 80% of the viruses I can think of either bind to cell targeting sialic acid on the cell membrane, or bind to cell targeting heparan sulfate proteoglycan. In fact, if I take a mimic of heparan sulfate proteoglycan, which is heparin, I can block many viruses: dengue, Ebola, herpes, papillomavirus, HIV, foot and mouth, West Nile, hepatitis B. But I will do that reversibly. But I can block a lot of them. So intrinsically, there's a very positive approach that these things are broad spectrum. Not only they are broad spectrum, but one can also start asking question: why is the attachment ligand target so conserved across viruses? Well, one reason that developmental biologists will give you is there has to be something in the virus that targets a part that is so conserved in the host that the host cannot mutate against the virus. But there is also another things that has always been in my mind that is completely unproven, but has always been in my mind. And that is that if you look at the viral replication mechanism with a thermodynamic eye, you see that the virus unpacks and repacks in the same cycle. So one can ask oneself, what is the spontaneous event here, unpacking or repacking? There are some virologists that will say that the unpacking is spontaneous, so that the packed virus is in a metastable state, and then when it goes on the cell membrane, it goes into an activated state that will later lead to unpacking. And later in the activation can be due to the pH in endosomes, can be due to enzymes that are there. But maybe, I'm just saying maybe, there is some coding in the binding to the cell membrane that starts this activation mechanism that will lead to the unpacking. Why am I saying this? Because with this in mind, I thought, well, there are many known compounds that are able to bind to viruses, but their reversible nature makes them difficult to translate into drugs. If I can imitate better the cell membrane, maybe I can activate this process and lead to the unpacking of a virus outside a cell. And I will then have an irreversible cascade of events. that starts with a reversible binding, but then is irreversible in its nature. And this irreversibility will actually make my compounds much better drugs and much better translatable into drugs. How can I do this? Well, I study in my research surfaces of proteins, and in particular, what is the role of exposed hydrophobics of proteins. And I know that the attachment ligands, so as well as the S proteins for that matter, have many exposed hydrophobic. And I also know that there's a lot of hydrophobic on the cell membrane. So I thought, well, a better way to imitate the cell membrane is to take these compounds, add a lot of hydrophobic to them, and maybe add them in a multivalent way, so in many ways, so that upon binding, what will happen, binding between the viral attachment ligand and the compounds that I'm going to design, there will be first binding in a preferred geometry, but then there's going to be later a strong and tight hydrophobic contact that will force the protein to go in a secondary conformation. And this secondary conformation will be an expanded one, and hence apply a pressure on the viral ligand. And this pressure will ideally lead to viral unpacking, or at least a virus going into a new conformation that is not the original one, that is not infective. If you go in the literature of these compounds here, you'll find a lot. Many that have gone to clinical trial and failed, in my opinion, because of their reversibility. I can expand on this. And you'll find, for example, gold nanoparticles. The king or the queen here is heparin. You make heparin-like compounds, and you'll have inhibition of viruses in vitro. You find gold nanoparticles, which is great for me because my group works on gold nanoparticles. And so what we did, we took the literature gold nanoparticles, and we simply took the hydrophobic part and made it longer. We knew these particles were non-toxic, which is a key design parameter whenever you go in antivirals. And we immediately saw that these particles were able, in a broad spectrum way, exactly like heparin, exactly like the literature nanoparticles, to block viral infectivity in nanomolar range, which is what you find in our literature. How do we do this experiment? We just simply take a fixed virus concentration, and we add many different concentration of our drug, and what we are plotting for you is the infectivity as a function of a concentration of a drug. And I'm quoting for you the EC50, which means the concentration of the virus, of a drug that blocks half of viral infectivity. Now, let's focus on the EC99 concentration, that is the concentration that blocks 2 logs of infection. And now I'm going to describe a very important experiment here. If we take a drug, and we're going to take here 2 exemplary drugs, a reversible drug, this would be the monoclonal antibody, for example, or an irreversible drug. And we take a control, which is virus alone, and a virus plus drug, okay? The virus plus drug is designed so that the drug is at the EC99, so it's blocking 2 logs of infection. Now, if we dilute both the virus and the virus plus drug many times, and we plot the dilution at various concentration, but here I show you at the last dilution where we can see a infectivity of a virus. What we see here is, if we've done the experiment correctly and we have a reversible drug, at the last dilution, the infectivity of a control, so virus alone, or of a control plus drug, will be the same, even though we started with 2 logs difference, because at that concentration we went below the binding constant, and the virus and the drug are detached. And so now we're going to have an infectivity. But if our effect is irreversible, no matter what we do at the last dilution, the difference we find is exactly is the same as the difference we started from. It's still 2 logs. Okay, this is called a virucidal test. It's a test that virologists do since the 60s. And it's a test where you plot this last dilution. Okay, that's the convention, starting from the EC99. So let me show you here what we found against in our experiments when we tested against HSV2, herpes simplex virus 2. We tested heparin, which is, as I told you, is a known broad spectrum antiviral compound that unfortunately has never been translated into an effective drug. When we tested heparin, we found that we had nanomolar efficacy, we had very good cell viability, but the effect was reversible. When we tested the nanoparticle that we had in the literature, we found same nanomolar efficacy, we had same cell viability, reversible effect. Just by taking the same nanoparticle, same ligand, making it longer, we don't do anything in the efficacy, nanomolar, cell variability the same, but the effect now is irreversible. And it's irreversible against herpes simplex virus 2, papillomavirus, respiratory syncytial virus, HIV pseudovirus, and dengue. We can do the reversibility test at different time points, and we see that the effect, the irreversible part of the effect, takes minutes to take place. And we can do cryo-TEM. And actually, this cryo-TEM will show us that the nanoparticle actually, at the end, I'm going to be more precise in a second, they destroy completely the virus. And this is why the effect is irreversible. Now, and the reversible nanoparticle don't do this. In the reversible nanoparticle, we don't observe this endpoint, basically. Now, we have done some, not we, a collaborator of mine, Peter Kral, has done some very nice simulation on the papilloma binding viral attachment ligand. But why papilloma? Because it's a non-enveloped virus. It doesn't have the membrane. And what he observes is a bit what my crazy experimentalist mind had seen. That is that the nanoparticle that have a long hydrophobic tail really enter inside the viral attachment ligand, and they do produce a stretching of this ligand. Okay, the other controls that were done by simulation showed no entry. You know, the problem is what happens with the enveloped one. This is still a challenge to understand, but in cryo-TEM we can see this. This is herpes simplex virus 1, and you see we can see the envelope. This is the capsid of a virus, and the black dots here are our nanoparticles. What happens is the envelope, with or without nanoparticle, tends to have spontaneous bulging. And the nanoparticle always concentrate at the neck of the bulge. And what happens here is when the nanoparticles are here, this bulge becomes long and always has a trailing of nanoparticles. And at one point, the capsid is expelled by the envelope, or vice versa. Now, the viral attachment ligand, in the case of express, it's still attached to the capsid, so the nanoparticles keeps attaching to the capsid. And at one point, this is a Z-stack, you see the nanoparticles break the capsid. And at that point, the inner part of the capsid is positively charged, and actually the nanoparticles get inside of, and they coat all of inside, breaking the virus. Now, in my research, we did all of this to have a clinical effect, to translate this. I'm going to be quick here, but we've done tests on human tissue regrown at the air-liquid interface, which is what we call ex vivo. And what you can see in this thing, that in co-treatment, so giving the virus and the nanoparticle together, in pre-treatment, nanoparticle before, virus later, or post-treatment, nanoparticle after 1 day after the virus, we can block the infection. If we use the reversible nanoparticle, we can start by VC99, so we can block the infection, but 2 days later, half of infection is back. 3 days later, the infection is completely back. And we actually have in vivo data where with this particle, we could clean the lungs of a mouse from RSV, respiratory syncytial virus. Now you can ask yourself, can we give a nanoparticle in the lungs of people that have coronavirus and maybe give it every week to people that go back to work? That's unlikely. So once we arrived at that point in my research, what I asked myself is, what have I learned? And what we had learned is that we needed to fight virus, long hydrophobic chain, terminated with sulfonic acid, which is this heparin-like moiety that attaches to many, many viruses. And we needed a lot of them. So what we did, we said, okay, we're going to use naturally occurring sugar called cyclodextrin that are used in many FDA-approved drugs. We're going to attach the same chemical group we attached to nanoparticle to their primary phase, and we're going to see if they work as drugs. Luck was that there were commercial compounds that looked a lot like we were doing, but we found lots of hydrophobic. Among them, an FDA-approved drug called Captisol, which is a solubilizer for chemotherapies. And these drugs block viruses, but in a reversible way. Captisol doesn't even block viruses, to be clear, while our cyclodextrin was able to block viruses in an irreversible way. This is HSV2, herpes simplex virus 2. Again, very broad spectrum drugs, very broad spectrum drug. We can do here a DNA release assay, and what we show is that when we give our drug to herpes simplex virus 2, we will actually release DNA, which means that we are irreversibly damaging the virus to the point that the inner part, the inner content of a virus is released. And again, Peter Kral did this time the calculation on the effect of cyclodextrin on the virus. These are reversible cyclodextrin, and this one is a reversible cyclodextrin. This is something that has a mixed effect. This one is the cyclodextrin that I showed you before. And what you're seeing here is the trimer that composes the attachment ligand for herpes simplex virus 2 after it has interacted with the cyclodextrin. And in the case of the reversible one, this is the distance between the center of gravity of the three trimers, the average distance in time. You see, when we give the reversible cyclodextrin, this distance is not affected by the attachment of the compound to it, but when we give the irreversible one, the distance grows with time, which is exactly what I had sketched years before. So you cannot imagine how happy I was when I saw this calculation. And also I have to say that this is due to the fact that many, many arms, in the case of the irreversible one, bind. And again, this works ex vivo, it resists mutation, and it has worked in vivo against RSV. I can tell you, this is not published, that we have preliminary data that it works in vivo against HSV2, herpes simplex virus 2, but preliminary data show that it works also against RSV, a respiratory virus. To conclude, I will just say that we have developed drugs that also work against the other family of viruses, which is the family of viruses that binds to sialic acid and does not bind to heparan sulfate proteoglycan. And in this family, influenza is the main target. So we have designed a cyclodextrin again, but has an hydrophobic tail and has an attachment ligand that binds to many influenza strains, among them a few that are pandemics, and it does it in an irreversible way. And in this case, we've taken the attachment ligand and we've attached it to cyclodextrin with an hydrophobic tail or an hydrophilic one, obtaining two drugs that had the same effect. Only one was irreversible, one was reversible. In this case, clearly, if we went and worked on tissue, the irreversible one showed no effect, but the reversible one, sorry, showed no effect, but the irreversible one showed good lasting coverage up to 6 days after. Now we have even a month after treatment. And this translated in excellent result in vivo. These are these results we obtained recently from an experiment in mice from NIH. And what I'm gonna highlight here, that this is the control sample where we had only 2 mice survive after being infected with a pandemic strain of influenza, and at the highest concentration of our drug, 10 out of 12 mice were saved, if you want, survived. This is Tamiflu, is a known drug against influenza that was given, though, at 10 times the concentration of our drug. I will stop here. I have to thank the people that worked on this 10 years project. This is the first collaborator of biochemistry, Silke Kroll, with whom we co-supervised these 2 PhD students, and then this postdoc took over. And the first virologist to collaborate with us was David Lembo, and Valeria Cagno dedicated half of her PhD to this project. Then she moved as a postdoc in the lab of Caroline Taparel, with whom I currently collaborate, and we've done and I've done all of cyclodextrin work with her. Peter Clark did the simulation together with Lela Vukovic. Marie has done the in vivo result. Paolo and Marie are in my own group, the students that dedicated their work to do the nanoparticle and to perform the cryo-TM study, and Sam is the inventor of cyclodextrins. This is my group right now, where Matteo has done all of the most recent work on cyclodextrin and is doing work on anti-coronavirus, together with Young, that has designed the anti-influenza molecule and new anti-influenza molecules. Francesca is developing new concepts to fight coronavirus, again with irreversible approaches. This is the key of what we do. And I'll stop here and take any questions that you would ask.
Hi. Okay, Francesco, thank you very much for this insightful presentation. And after these 2 nice presentations, we will now move to the last part of today's webinar, which will be the part where we will be dealing with the questions that we have received from the attendees to Francesco and Eric.
Should I start, since I had a chance to look at the questions while Francesco was speaking?
Absolutely.
Yes.
So I got a couple of them sent over in a Word document here. So the first one is whether the membrane composition is known. So this is not my expertise, but thank God we have a really skilled group of people in Stockholm sitting in, partly live tweeting on our intranet, but also looking at membrane composition. So Sergio Canesa in Lucy's group has been speaking to Seema Khalid about this in particular, and we know a little bit of it. So according to Seema, it's kind of a mix of PC, PE, PS, PI, and cholesterol. The caveat there, though, is that all viruses they don't produce lipids themselves, right? They get this host cells to do the hard work for them. So I guess one question is, what is the lipid composition we end up with because we're getting it from the host versus the lipid composition that is functionally critical for the virus? And the less I know, much less about. There are some indications that cholesterol is important for the ACE2 binding because the ACE2 would tend to occur in the cholesterol-rich regions of the cells. So we know, yes, it appears to have a little bit of a role, but exactly how important the specific composition of the viral bilayer is, we don't really know, or at least I don't know. We also had a question about the spike glycoprotein, the sugars, what their impact of the ACE there is. And same caveat there. I don't think it's the ACE binding. I don't think that the glycosylation is critical for that. But I think there are indications that the glycosylation pattern is important to get the transmembrane serine proteases of the host cell to identify the spike protein and then cleave it. And this cleavage is then what exposes the fusion peptides and everything that causes the actual infection of the host cell. So the glycosylation pattern is critical. Mm-hmm. How much do we know about the membrane channel forming proteins of the virus? Oh, that's a good question. We know a little bit about it. There was a related question about that, about operating frame 8 versus 3A of SARS-CoV and SARS-CoV-2. We know that there are some channels. They are believed at least to form channels in the host cell. So they're somehow involved in the infection process. I don't think there are any channels in the virus itself, because remember, a virus doesn't really have energy turnover outside the host cell. But I think all I know about it is that they're likely to be involved in the infection part. And we had some questions about cholesterol and the viral lipids. So we do believe that there is cholesterol there. We don't think that there are any lung-specific lipids, such as surfactants or anything in the virus. But again, don't know is not the same thing as nobody else knows. I'm not aware of it. Mm-hmm. There were some questions about how reliable the usage of cell lines is to get the virus host cell interactions compared to, say, primary lung cell cultures or so. Again, this is not my expertise, but I would argue not at all, but it doesn't necessarily matter, right? Because you start out with the cell, and the first thing you test, if you have the wild type virus, can you infect the cell? If you can't infect the cell, then it's pointless as a test. But if we can get this virus to infect the cells and it produces RNA, so then something is definitely happening. If something is happening, I can now test by adding an inhibitor and see if we stop it happening. And sure, there's theoretically a case that this particular inhibitor will not stop this in this particular cell line, but it would stop it in an actual lung tissue or something. But I would say that that likelihood is so low that we ignore it. So that if we see something happening in a cell line, there is a small chance that it might work in tissue too. But if we don't see anything in a cell line, the hope that it would work in real tissue is so small that we ignore it. But sure, in theory, it could happen. Mm-hmm. There were some questions about the first viral responses related to large conformational changes triggered by ACE2. It's not large is a relative question, but yes, there is a conformational change. And whether we could inhibit this. And I guess the two approaches followed there is that one can try to somehow inhibit the ACE2 binding, or somehow get an antibody to either get your immune system to fight the virus, although that comes with the caveats that I mentioned at the beginning of the talk. Or the third part is that can you somehow inhibit the cleavage? Because if you inhibit the cleavage, sure, you might get it to bind to ACE2, but it won't really infect your cells. But the spike protein will be important. we see there were some more general questions about the sizes of these proteins related to MD. So the size of these proteins, the main protease is roughly 30 kilodalton, if I recall correctly. So around 300 residues, a few thousand atoms, maybe 5,000 or so. So not tiny, but certainly not large compared to large human, multi-unit subunit ion channel or so. It's certainly possible for MD, even on a workstation or GPU, to run multiple microseconds of simulation with this. But the caveat again is that, can you get a simulation to sample multiple states? Do these states correspond to actual different apo versus holo states? Because it might be that the protein won't really assume a state corresponding to the bound state until you have the actual ligand bound. And if you don't have the ligand bound, it might only spend 0.1% of its time there. And that's why people try to attack with methods such as Markov state models or so, to not just run one long simulation and wait for the conformational changes to happen spontaneously, but somehow try to force the simulations to map all of phase space, including the regions that are less favorable under wild type conditions. And there were lots of good questions here about what one could do, say, with QMMM, because that I cheated a bit here. I didn't go into detail. So even some of these inhibitors are covalently bound, right? And if you want to measure, if you want to study the covalent binding or something, classical MD is not going to do that for you. We also know relatively little about how they are. Well, the cleavage itself will also require QMMM. On the other hand, I don't think that these are interesting problems, but they're not necessarily the most important challenges for us to be where simulations could aid drug design, because we know that the cleavage happens. We don't necessarily need to simulate that because it's already known experimentally. We know what the binding site, and in general, people know enough about these mechanisms for how the binding sites and the actual entire cleavage enzymatic reaction happens. So I think if one wanted to do QMMM here, one should have a slightly more specific question. So what is the question you're trying to answer? And just running a QMMM simulation is not the question, just as running a classical molecular mechanics simulation is not really a question either. Mm-hmm. My hunch here, where I think MD simulations can be useful, is that again, map out the different conformational states that these proteins tend to assume, to see, can we get something to bind in a transition state, or can we somehow inhibit things? But rather than having the MD simulation be the end goal, in many cases, I would guess that what I could hope to get from MD simulation is having, say, 10 important conformational clusters. And then rather than just docking to the experimental PDB, I now have 10 different clusters I can run docking to. And I just might be lucky that they'll help me to pick up something that I wouldn't see in docking otherwise. But these are fairly long shots. In general, I think good experimental assays is going to be more important than anything else. But that's, of course, that's when it comes to developing treatments. Then I'm also a scientist, and it's a super interesting question to understand, for instance, how these ion channels work, what are the roles in the entire life or infectious process of the virus and everything. So there are many questions here that are important and useful to answer, even if they might not be the ones contributing to an antiviral. Mm-hmm. I think those were most of the questions. Sorry if I missed any.
Yeah.
No, indeed, Eric. I think that these were most of the questions that were asked. I actually have a couple of questions for Francesco. So you mentioned the issue of delivery, and I was wondering, how critical is it? So of course, it's critical for turning anything that works in a lab into anything that is then going to be an effective drug. And how far away are you from actually getting there? What are the main criteria that you need to meet?
Oh, very good. So in terms of delivery, that's the really big problem they will have to make a true broad spectrum antiviral. Because when I tell you that my drug works on herpes and RSV, and I don't want to, maybe also coronavirus, let's put it like this. The problem is that getting to the lungs and to reproductive tissues is very different, okay? And so delivery is a key thing, okay? Now, when we talk now, because now in these days is very trendy about possible epidemics, those for the most part are respiratory viruses. And in that sense, that's actually easier from a virological point of view, because the organ that is mainly targeted is the lungs, or the whole respiratory tract. And there you can go directly from the mouth or from the nose, directly there. And that's what we are developing. We are developing it. We have good data on that. We know we can get there. When you talk about how far I am, the real big problem here is my discovery is if I add enough hydrophobic to something that can take me to a virus, I will irreversibly inhibit its infectivity, which I can simplify and say I can destroy a virus, right? The problem is when I add that hydrophobic, am I gonna become too toxic? And toxicity is a black box nowadays. The only thing I can do is to try it, and I have to try it in many animals before I can try it in a human. That's where I am. I am actually trying to establish protocols to efficiently look at the toxicity of this compound. Also, your question allows me to say something that, you know, what I've discovered is a generic thing. It will not just, if it works in something so different as gold nanoparticles and cyclodextrin, it will work on many, many things. If Eric gives me something that can take me to the S protein, I will add enough hydrophobic, and I'm quite sure that I will irreversibly block coronavirus, right? Will I do it in a non-toxic way? That it needs to be tested. But if it's toxic with cyclodextrin, maybe I can put it on another scaffold and make it non-toxic, unless there's something intrinsic there, right? So I have a generic design rule on how to, once I get to a virus, inhibit it in a permanent way. And that's something that I think a lot of people that have designed antibodies or things like that, this may be a limitation on the study that Eric showed for SARS-CoV-1. The reversibility of the effect has never been looked in depth, but dilution in the body is an inevitable phenomenon. You will always dilute the drug or the virus or both.
Mm-hmm.
Thank you. And then a second question, perhaps. I mean, what kind of questions would you like to ask to people who do simulations that we cannot answer yet?
My dream is in one of the main limitation in my measurement. I've shown you that I can measure the reversible part and quote you a number, nanomolar. So we all know how to do that. When I go to an irreversible effect, how do I quantify that? What I would like to have help in doing that, because I've shown you one molecule, but in my lab, you can imagine we have many molecules that work. And they work very well, very, very well. But what works best? I don't know. When we go to the reversible part, I have a ratio that is very important, which is the EC50 divided by the maximum toxic dose, right? And that gives me, what number do I put when I actually have to quote the irreversible part? Because there's something hidden in my talk, which is paradigm breaking for virologists. My nanoparticle work with an IC50 that is 3 orders of magnitude better than the cyclodextrin. Yet when I go in tissues and when I go in vivo, there is no difference between the two. The cyclodextrin is as effective as that. And I think I know the reason for that, because if I try to measure the time scale of irreversible effect in the cyclodextrin, it's seconds, we know it. If I try to measure it for nanoparticles, it's minutes, as I showed you. So irreversibly, I know that the cyclodextrin are better. But how do I measure it? And that, if for simulation people, if they could show me, or even help me think, how do I need to design my molecule to be better in the second step, which is the irreversible effect, the hydrophobic contact? Can people simulate that? Can they help me? That would be fantastic. Because if I can do that, if I find that my aliphatic chain is too toxic, I will change design and go to the next one, right? Now I know that irreversible helps me a lot in vivo, which is the only thing that counts here. So can you guys help me in understanding the cascade of events and designing the most efficient one?
Okay.
Well, I think it's a challenge for us all. Ignacio, I didn't have time to follow the question feeds.
I've seen that recently they have arrived also a couple of additional questions for Francesco, a bit more specific. So one is about what information is known about the metabolism of these cyclodestrins in vivo and how that's affected by the new ligand.
So, and the two questions have opposite answer. Information about the metabolism, it's all known because there's like 30, 40 FDA approved drugs where the cyclodextrin typically are solubilizing agent. So a lot is known. Very little is known when the injection mechanism is the one that I'm talking about in the lungs. But basically, these things have only one problem. At high concentration, they have side effects on the kidneys. But we think that from something that is in the literature, that when we go from the lungs, most of the molecules will be enzymatically cleaved before it goes in circulation. The second part instead is the opposite answer. What is known when I add my hydrophobic tails? Nothing. We are studying it now.
Okay. Maybe one last question because we are running out of time, but there is this question about the relevance of these long hydrophobic chains and on the virus, how much do they depend on the micromolecule and nanoparticle they are attached to?
So as far as all of the data we have, and here I'm talking about 10 years of data, so they are the fundamental part to make the effect irreversible. But as long as I don't know how to measure the efficiency of irreversible, which is what I said before, I have 2 logs reduction in all of our viruses where it works. If I go change them in terms of length, not much changes. It changes in terms of solubility, and that's key for toxicity later. So we kept it as short as possible. Longer, it should work better if I were to measure it, but then I have toxicity effect down the road. Across viruses, not much is changing. If I make the chain less flexible, I lose a lot of the effect. So instead of 2 logs, I get 1 log. So it needs to be flexible, flexible and hydrophobic. A challenge for the computational people to show me exactly how it works.
Yes, I think that's part of the goal of these webinars, to bring these issues on the table to a broad audience and to try to show where challenges are. So hopefully this event will contribute also to the research along these lines.
And for anybody, if you have, for me, small peptides, small molecules that can take me to the coronavirus, I'll add the irreversible effect and I'll test it for you.
Okay.
Take me there, I'll take care of the rest.
Right, that's a specific question for the audience. So let's hope it's perceived properly. So I think it's a point here to stop this very engaging session. So thank you very much again, Francesco and Eric. I know these are particularly busy times for you, but you took the time to be here with us. So thank you again. And thank you to all the people attending. As I mentioned, the presentation, the whole session is recorded and is available on the CICAM website. And I invite you, I hope to see you next Tuesday. We'll have another session. This one will be around this project that Eric mentioned earlier about Escalate and what they are doing also in the context of the COVID challenge. So thank you very much. And
Thanks
hope
a lot.
to see you again. Yeah, thank you.
Bye.
Yes. Bye. Bye-bye.
Bye.